Dear learners, hope you all remember that we have learned about carotenoids. Today's module focus on yet another important polyphenol compound, that is anthocyanins, which is a coloring pigment used extensively in food products. Let us begin. Let us understand what is anthocyanins. In acidic condition, anthocyanins looks as a red pigment, while blue pigment anthocyanins happens in alkaline condition. Anthocyanins is measured as one of the important flavonoid, although it has a positive charge at oxygen atom of C ring of basic flavonoid structure. Anthocyanins are normally found in most of the fruits and flowers. The most red, purple and yellow colored flowers control anthocyanins. The red flowers are red hibiscus, red rose, red pineapple sake, red clover and pink blossoms. These red flowers are edible. The blue ones are cornflower, blue chicory and blue rosemary, and purple ones are purple mint, purple passion flower, purple sake, common violet are found in lavender flowers. All these flowers are edible flowers. Some of these flowers have been traditionally used as a folk medicine as a colorant in the food. In addition, traditional usage, red, purple and blue colored fruits are commonly consumed for their favorable effects. The color pigment of anthocyanins from berries, blackcurrant and other type of red to blue color fruits are strong antioxidant. Moreover, anthocyanins rich black carrot, red cabbage and purple potato are prospective functional foods that has been consumed for the prevention of disease. Learners, let us now understand the chemistry and structure of anthocyanins. Anthocyanins occurs principally as a glycosides of their respective glycon. Anthocyanins chromophores with the sugar moiety generally attached at 3 position on C ring or 5 position on A ring. There are about 17 anthocyanins found in nature, but only 6 anthocyanins which are commonly used are cyanidine, delphinidine, petunidine, peonidine, pelargonidine and malvidine, with cyanidine being the most common and universally spread and greater importance in human diet. The diversity of anthocyanins are due to number and position of hydroxyl and methoxy groups on the basis of that anthocyanidins frame. The identity, number and position at which sugars are attached, the extent of sugar acylation and identity of acylating agent all are important. The intensity and the type of color of anthocyanins is affected by number of hydroxyl and methyl groups. If hydroxy group is prominent, then the color goes towards bluish shade. If methoxyl group is prevalent, then redness is increased. Anthocyanins is a derivative from flavanol and it is the simplest structure of flavium ion, that is the absence of ketone oxygen at the 4 position. The empirical formula for flavium ion of anthocyanins is C15H11, where C stands for oxygen, H for hydrogen and O for oxygen, with a molecular weight of 207.24 per gram MOL, that is milliliters. On the other hand, anthocyanins are glycosylated form of anthocyanin results in blue and purple color plants. The pH determines the stability of anthocyanidins. At low pH, that means at acidic condition, the colored anthocyanins are present, whereas at higher pH, the colorless chalcone are present. Anthocyanins are less steady at greater solution temperature. Learners, hope you have understood the chemical structure of anthocyanins. The common sources of anthocyanins in our food is red onion, red cabbage, cranberries, raspberries, blackberries, red and black grapes and blueberries. Anthocyanidins, which is found as a commercial product or as a functional food. Let us find out that how. Anthocyanins extracted from plants have been used as a food additive. The common food additive, that is E163, is one of the most profitable additive derivative from fruit anthocyanins such as grape skin. It is a purple food additive used for making purple colored jam, confectionery and beverages. Recently, synthetic food dyes involved public concern concerning the safety and the adverse effect on human health, mostly neurological function and behavior effects. The use of anthocyanin based colors in yogurt drinks and some mixed fruit juices is becoming more popular. Some companies did use the synthetic dyes in their product. However, these synthetic dyes may be toxic if consumed in excess amount. Recently, acylated anthocyanins are a food colorant used in the food industry due to their high stability over non-acylated anthocyanins. Learners, let us now find out about the bioavailability, absorption, metabolism of anthocyanins. In vitro studies on antioxidant action of anthocyanins are plentiful. On the other hand, after ingesting the food in which these pigments are present, they vary, perhaps metabolized in the organism with the probable variation of activity. The information on absorption, metabolism, tissue and organ distribution and excretion of anthocyanins in human subject is very limited because they are complex, expensive, lengthy and sometimes with the contradictory results. Anthocyanins glycosides can be quickly absorbed from the stomach after consumption and they enter systemic circulation after passing through liver. Methylation and glucuronidation responses can occur in this organ and some of the metabolites are transported to intestine as bile. Anthocyanins glycosides which were not absorbed from stomach passes into small intestine converting to a combination of chalcone and quinonadiol forms due to pH of this organ. Further, the absorption seems to take place in jejunum, being the transport mechanism is unknown. Absorbed anthocyanins move in systemic circulation after passage through liver and may be metabolized. Anthocyanins that reach the colon are exposed to microbial population able to transport them to sugar and phenolic compounds with further degradation by disruption of C-ring of yield phenolic acid and aldehydes. Learners, let us now learn about the health benefits of anthocyanins. The anthocyanins and the cancer. The anti-cancer activity of anthocyanins from many fruits and vegetables have been demonstrated to inhibit the initiation, promotion and progression of several cancers such as breast cancer, prostate cancer and liver cancer. Extensive investigation have been performed to determine the molecular mechanism of anti-cancer activity of anthocyanins. with the result indicating that anthocyanins can inhibit several signaling pathways which involved in tumor growth and apoptosis. Anthocyanins have also been found to be helpful in nervous system. Human brain is susceptible to oxidative damage. Brain damage is due to peroxynitrite nitration produced by tyrosine remains that is present in enzymes and proteins. Nerve receptor sites are obstructed by nitrite tyrosine. Active element pelargonidine in anthocyanidin prevent the nitration of tyrosine and restrict incidence of any neurological damage caused to the nervous system. Anthocyanidins have also been found its role in cardiovascular diseases. Anthocyanin-rich fruits and vegetable could effectively reduce the risk of atherosclerosis by improving endothelial dysfunction, inhibiting oxidation of LDL formation and promoting macrophage, which reverse the cholesterol transport. Anthocyanins are very effective in reducing the stress on blood vessels. Microcapillary integrity is maintained by anthocyanidins. The capillary walls are stabilized by anthocyanidins. It is very effective in fighting ischemic reperfusion. In this condition, reduced oxygen supply creates the oxidant that cause white blood cells to stick to capillary walls and increase the wall permeability. This condition can be improved by taking fruits with high anthocyanidin content. Let us conclude, learners. Anthocyanins are one of the most wide-range families of natural pigment in plant empire. Their health favorable effects have been recognized in many vivo and vitro studies. It appears that several signaling pathways including mitogen activated protein kinase, nuclear factor, AMP activated protein kinase, as well as some crucial cellular process such as cell cycle, apoptosis, autophagy and biochemical metabolism are involved in helpful effects and may provide potential therapeutic targets and strategies for their improvement of wide range of diseases in future.